The polymerase chain reaction, like gel electrophoresis, is a widely used method in molecular biology. DNA and RNA sections can be qualitatively detected and quantified in a targeted manner. The polymerase chain reaction is divided into the so-called classic PCR, qPCR, and FRET-based real-time PCR. Let's first talk about classic PCR. For each form of PCR, the DNA must first be extracted from the material to be examined. To do this, cell walls are broken down with an enzyme and the DNA is made visible as a cloudy clump using sodium and alcohol. After washing with alcohol, we get pure DNA that we can dissolve in water. The spiral DNA strand must be despiralized for further processing. By heating the DNA at 95 degrees for 10 minutes, the DNA is, so to speak, smoothed. This means that enzymes and other substances have better access to the individual building blocks of DNA, especially the peptides adenine, thymine, guanine and cytosine. By heating the DNA above 95 degrees Celsius for a little longer, the hydrogen bonds between the peptides are broken and the DNA double strand splits into two single strands. After the DNA has been cooled to 60 degrees Celsius, so-called primers are added to the DNA. These primers are short DNA sequence sections that attach to specific locations on the DNA. Every single gene and every known mutation can be identified with a suitable primer. In addition to the primers, plenty of peptides are also added, i.e. the aforementioned adenine, thymine, guanine and cytosine. In the further course, the DNA single strands should be completed again into two double strands. This requires a special enzyme and the enzyme used here is called DNA polymerase. This works best at a temperature of 72 degrees Celsius because it comes from a heat-loving bacterium called Thermophilus aquaticus. The enzyme is therefore also nicknamed Taq polymerase. The DNA polymerase binds the freely moving peptides, which we added earlier, with the bases of the single strands. As in the original DNA double strand, guanine and cytosine, as well as adenine and thymine, are contrasted here. From the respective primer, the individual strands are gradually completed. So far we have smoothed our extracted DNA, split it up, identified individual DNA sections and from there completed the two DNA single strands into two DNA double strands. By repeating this process over and over again, we can potentially generate an infinite number of DNA double strands from a single DNA double strand. This enables us to carry out further studies on the genetic material. In order to see whether a particular gene is actually present and how many DNA strands we have generated, we first have to make the genes we are looking for visible in the DNA. Primers are used again here. Similar to before, the primers bind to their corresponding DNA sections. As soon as the primers bind to their specific DNA section, a dye linked to the primers can be activated. This is usually a dye that is activated by exposure to UV light. The primers bind to the gene, the dye on the primer is activated and the gene we are looking for, if it is present, becomes visible to us. With this method of classical PCR, typical genes of viruses, bacteria or hereditary diseases can be detected.